Have you ever wondered how genetics might play a role in language translation? Picture a world where a single gene, the hypothetical linguistic gene XZ, holds the key to this fascinating realm of study. Imagine a gene so intricate it encodes a protein complex enough to facilitate the role of linguistic translation. This protein, a masterpiece of biology, would likely consist of various domains or regions, each with a distinct role. Some of these fragments might be involved in protein-protein interactions, others in DNA binding, or even enzymatic activity depending on their function in the translation process. Now consider the potential function of this protein. In the vast landscape of the human brain, certain regions are associated with language processing. The Broca's area, responsible for speech production, and Wernicke's area, involved in understanding speech, are two such regions where this protein might regulate gene expression. Alternatively, it could be a critical player in the modulation of neurotransmitter systems, such as dopamine or glutamate signaling pathways that influence language-related cognitive functions. And where would you find such a protein? It could be localized to specific subcellular compartments within neurons, such as the nucleus, cytoplasm, or synapses. Here, it would exert its effects on the molecular machinery involved in translation. Yet, the expression of this gene and the activity of its protein product would not be random. It would likely be a finely tuned process, regulated in response to various internal and external stimuli. Regulatory elements within the gene's promoter region, as well as post-translational modifications of the protein, could modulate its activity in a context-dependent manner. Moreover, this protein would not work in isolation. It may interact with other proteins, RNA molecules, or regulatory factors to form functional complexes involved in the translation process. These interactions could be critical for coordinating the complex molecular events required for effective linguistic translation. To summarize, the hypothetical linguistic gene XZ, encoding a complex protein, could play a central role in the activation of linguistic translation. It might regulate gene expression in brain regions associated with language processing or modulate neurotransmitter systems that influence language-related cognitive functions. It could be localized to specific subcellular compartments within neurons and its activity precisely regulated. And lastly, it might interact with other molecules to form functional complexes, orchestrating the intricate process of linguistic translation. While this gene remains a subject of speculation, the exploration of such possibilities broadens our understanding of the intricate relationship between genetics and language. It gives us a glimpse into the future of linguistics, where the mysteries of language translation may be unraveled at the molecular level.